Okay, so now we will go into the detail of the database, and you will find it available on the Design RESPIRE design website, sorry, web server, and the name is the RESPIRE here. So it's easy to find Design and RESPIRE. So you have a short description of what is the database. The link to the article is provided here, and we will go rapidly into the tabs provided. So first, I will recommend to go to the documentation, where we have tried to add many comprehensive information for what you will find. So the tabs here described for many different parameters. And if you go now on the erythropoiesis, the data processing tabs, the structural information, you will get details of what we did to select one template or the other one, etc. What is the database organization? So it means a lot of information put and assembled to quite logically, or try to be logical on that. Some questions that we have got from reviewers or users. And of course, do you have the right to reuse that? Yes, we want to have this information used as much as possible. So don't hesitate to do that. This is meant for being reused, and this is the license also which is used by Uniprot. So this is knowledge. It is meant to be shared. Please cite us. But other than that, it's of course important to do. So I will go, for instance, here on the quick search tab. And as I told you, there are membrane proteins, and these membrane proteins are quite differentially labeled by Uniprot and the Gene Ontology Consortium. So if you click on this first list, you will get 417 proteins. And if you click on the other one, you will get another definition, and we will get a bigger list of that. So you can click on the list to see the difference if you want, but you can click on any value here to get a short description on that. For instance, HAC protein. And if we have a model for that, you get a 3D here, which is indicated. So I won't go into these ones. They are not interesting. I will go, for instance, okay, in the so you will get all this here. You can download it. If you go now on the other criteria, you can find in OMIM, if you want to see, is there a protein involved, for instance, known to be involved in transfusion? You can search for that. I won't restrict here. And there's one which is known to be involved in a transfusion accident. So you can click here to get the detail on OMIM. Okay. And we know that integrin beta 3. So you can click, for instance, you can change, sorry, you can search for that. You see, so you get integrin beta 1. And so you get the detail of one integrin. You see it was updated in February 2020, this description coming from Uniprot. Is this protein important? Yes, it was found in many different organisms, different publications. So are we sure that this information is available from many different publications? Yes, it was published in many different surveys. So you can go on the detail of publication if you want. Do we have information about its expression? Yes, if you take all the information from different stages of the cell differentiation, you see that the expression is increasing, then decreasing at the end of the process. You will get information about the sequence conservation with the color code from blue, not conserved, to red, very conserved. You can download a lot of parameters, so the protein sequence, the protein coevolution profile, which is important for prediction, the multiple sequence alignment, which is used to produce this graph. And since it's a protein, you can get also the gene ontology or the membranous definition of these proteins as a file, an independent file here. The domains found by Interpro is containing a lot of subparts. For instance, if you go on this one, you will get all the details here. I won't go specifically on each of them because you will go to another website. Do we have a structure? No, you see, no model was available because we don't have a lot of templates. We don't need to produce here. It's indicated because we have one structure. So if you click on that, you will display the structure and you'll see that, yes, we have a completely regular structure. So we have a lot of information about that. And there are many, many other structures describing this entry. So there was no need to produce a model. We have sufficient coverage from experimental data. What are the interactions? So here you can drag from this integrin blah, 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 integrin beta 1 here. What happens? And when you over here, you see this one is thrombospondin here. This one is galectin 3 binding domain. And you get a lot of other interactions for many, many proteins. So you can arrange the graph if you want to get a broader overview. You can do a screenshot of that. What are the gene ontologies associated with this process? Many of them, it's the case for many proteins. Surprisingly, it's many, many different proteins. And if you want to see, for instance, so I will just go I will come back on this later. If you want to see the genetics now for one entry, you will go to the description done by done by OMIM, the Online Mendelian Inheritance in Men. And you see many, many information there, and you can click on the website to get more information. I won't go again here. You see that this was revised this year with an update probably coming from Uniprot information. So you can register to this, and if you are interested in one protein, if you click on that, you will get the information only for this protein and not for the whole database. And again, the links for all the data. So if you go inside the other tabs, you will get the statistics. So I showed you already the documentation, but how many proteins are involved, how many proteins are, where are they shown, what's the average size, how many references you see, it's nearly 30,000 now. Many proteins have been updated and have restrict some of the criteria for models. So we have, we'll revisit this to have more models. When you click on just this, it will highlight what is the details. So it's cytoplasm. You see many, many different compartments defined by the gene ontology. And when you click on one entry, it will give you all the proteins where this blood microparticle, for instance, is all the proteins known to be inside this compartment. So you see it's very detailed. Again, I won't go into all the details. And the database is updated regularly. So you see now quite huge list. It was started publicly available in 2018. And the last update was done 1 month ago. So this is all for the recording. You can go there to have a look at the database, and please have a look at the article here where more details are provided and more insights into the predictions are indicated, and please cite us.